Welcome, everyone. The mission of NewLimit is to radically extend human health span, and we've been hard at work over the past year. We've got some exciting updates for you today. Now imagine with me just for a moment, what if you had the immune system, the mental ability, the metabolism even of a younger person? I think that all of us in this room would be a customer of that product if it existed, at least at a certain age. It would also be one of the most important achievements ever for humanity. And NewLimit was founded with this observation that all the major diseases that take us out later in life, whether that's cancer or cardiovascular disease or diabetes, these are all highly correlated with age. And it makes sense intuitively because as we get older, our cells lose function. They're unable to recover from damage or insults, or maybe the errant cancerous cell that appears when we're 20, our immune system is able to correct it. When we get older, maybe it's not. And so what if we were able to actually restore function that our cells had when they were younger? Could this actually be a way to go fix the root cause of many of these diseases? Now, typically, the largest companies, at least in recent memory, they've all been tech companies that are now trillion-dollar companies. We're seeing an interesting phenomenon just more recently, which is some biotechs are achieving these kinds of valuations, and they're doing it with a new type of drug. We call these trillion-dollar drugs after the GLP-1s, which have added about $1 trillion of market cap to Eli Lilly and Novo Nordisk. And the reason why they've gotten so big is that healthy people are taking them, not just people with a disease, but eventually a large percentage of the population. And the GLP-1s have now treated about 1% of the world, if you can believe that. It does seem to be no end in sight. We believe there are more trillion-dollar drugs out there, and in the aging space, the TAM is just so enormous because everybody over a certain age is going to take one of these drugs. So how might it actually be possible to tackle something this ambitious in the world? Well, you all know about genetics, so all of our cells have roughly the same genetic code, but our cells are very different between a cell, a skin cell, a muscle, a neuron, and this comes down to epigenetics, basically which genes are turned on and off in any given cell, which has wildly different outcomes and expressions for that cell. And Shinya Yamanaka, in 2012, he gave us this beautiful existence proof that changing age is possible. He found these 4 transcription factors, which are just genes that turn on and off other genes, those switches I mentioned on the prior slide. And he took an old skin cell and reprogrammed it to a young stem cell. So he moved it along the x-axis, changing the cell age, but also the y-axis, changing the cell type. A remarkable discovery that advanced the epigenetic reprogramming field. And NewLimit is trying to do something that's about half of what Yamanaka did. We're trying to change just the cell age on the x-axis without changing the type of the cell on the y-axis. Now, to do this, we're going to go search for novel transcription factors. We're not going to use the Yamanaka factors that he found, those 4. So we're looking for new ones. How difficult could it be, right? Turns out it's really, really difficult. There's about 10 quadrillion, that's 10 with 16 zeros after it, possible combinations of transcription factors, conservatively, that one could go look at that might reprogram a cell. And this would be very difficult to do in any given normal situation. That's far more than we could exhaustively search in our own lab. It's actually more than all of humanity has done in every lab for all time, cumulatively. It's far bigger than that number. So the exhaustive approach doesn't work here. Luckily, we are all living through the birth of AI. And you're going to hear later in this presentation about how that's allowing us to make this problem tractable for the first time and has allowed us to develop what we believe are the most advanced AI models for epigenetic reprogramming. Now, the company got started in 2021. We thought this might be a 5 to 10 year basic research project to get any sort of foothold on this problem. It's turned out to have gone faster than we thought. And in 2023, we did the first large-scale screen with young and old human donors. In 2024, we were able to find the first drug payloads that made human cells look younger phenotypically. And then we said, okay, they look younger phenotypically. Let's actually put them through their paces in functional assays, see if they actually act younger. And in 2025, at our last progress update, we announced for the first time we had found drug payloads that can reprogram old human cells and actually make them act younger. So now, in 2026, we are getting ready to turn these into medicines and get them in the clinic. And with that, I'm going to bring up the other co-founder of NewLimit, Jacob Kimmel, to tell you about some of our milestones in 2026.
Hey y'all, thank you so much for being here. It means a tremendous amount to each of us. We will try in the next few minutes to make it worth your while. As Brian alluded to, when we started the company, we thought all of this would take much longer. And in just the past year, we've had 3 big unlocks that I never expected we'd be able to talk about today. And I want to walk you through those really in the story of how one discovers a medicine and then turns it into an actual drug in human patients. The first big unlock in the past year was we were able to build AI systems that really doubled our rate of discovery. For every dollar we invest, we get twice as many payloads out that have the pro-youthful effects we're excited about. Based on the back of that success, we started our 3rd therapeutic program, now focused on the endothelial cells that line all the blood vessels throughout your body, really playing an important role in every tissue. And then finally, potentially most importantly, we declared our first preclinical candidate. This is the first medicine you can hold in your hand that we want to take forward and actually put into human patients. Now, diving into a bit of the details on the discovery process, we'll start in the same place that the story of every medicine does. NewLimit was really built not just to commercialize one particular product, in-license one piece of IP, and then wrap ourselves as a company around a single asset, but rather to build the technologies that would allow us to discover aging medicines reproducibly, one after another. We call that technology our discovery engine, and it's fairly easy to explain. We start by finding differences in young and old human cells. In practice, this involves actually going out and gathering those cells from young and old tissues from hundreds of different people. Then we generate genomic profiles that allow us to determine which genes are the old cells using, which genes are the young cells using. And they really give us a map then of what aging actually means molecularly in a given cell type. We then build AI systems that Brian alluded to that can help predict which groups of these transcription factors might actually make that old cell adopt a more youthful state. And those predictions are only as valuable as the data that they yield in the actual laboratory. So we take the most promising of them and test them in large-scale experiments. We've built some chemistry that allows us to test hundreds to thousands of times as many of these payloads as have ever really been tested before. And those 2 technologies work in a harmonious loop. As we run new experiments, we take that data and train better models. Those models then make better predictions and allow us to gather more valuable data. And over the past few years, we've run this loop over and over again. And we've cumulatively generated something like 1,200 times as much data in this regime as every other group in the world combined to our knowledge. Now, that data has allowed us to build what we believe to be the best models as well, that can now explain more than half of all the variation we see in experiments before we ever go into the laboratory. We've managed to take a few of the most promising of those medicines we've tested and carry them forward and turn them into actual prototype drugs. At this stage, we now have dozens of prototype medicines we've been able to generate that can restore youthful function in old cells. Now, I want to dive a little bit deeper into how this technology actually works and how we're able to make those discoveries, because I think absent some of the AI systems we've built, it's very unlikely we would have been able to find anything interesting in this regime. So just as Brian alluded to, there are far more combinations than we'll ever be able to test experimentally. Doesn't matter how hard you work or how smart you are, you're going to run out of space on the surface of the Earth before you ever actually test all these combinations. And so we started by building models that could actually predict from just the description of some of these transcription factor genes that are able to remodel the epigenome, what will happen when we turn them on in an old cell. And that might sound pretty simple, but the first thing we actually had to solve was figuring out how to represent these transcription factors in a way that a machine learning system can actually reason about. And here we can stand on the shoulders of giants. We can take advantage of some emerging foundation models, both of nature's languages, like protein sequences, as well as human natural languages, like large language models almost all of us have used. And together, these systems can actually summarize everything we know about these transcription factors a priori. All the human research that's been done, everything that's present in their natural sequence biology, into a single numeric representation. We call this an embedding. And I like to think about this as a distilled numeric piece of knowledge so that we can build our models on top of a lot of the intelligence that's already been accumulated. We then train systems which can predict from those embeddings what turning on groups of those transcription factors might do to an old cell's state. Will it make it look younger? Will it keep it the same cell type? Will it turn it into another cell type? Or really any other property we're able to measure. Now, whenever anyone introduces any technology with a bunch of cartoons, you need to ask where the figures are, because otherwise they might be trying to sell you snake oil. So here are the figures I have the privilege of presenting that our AI scientists are able to generate. And what I hope to be able to convince you of is that these systems can now predict with really high accuracy what actually occurs in the laboratory before we ever go run an experiment. They can explain the vast majority of the variation. Now, the systems are not perfect. I'm not going to claim that we can zero-shot our way from just a prediction in silico to a drug we want to take into human trials, but they are far better than I am at prioritizing what to test next. And so once we have these systems trained, we can actually turn them around and try to use them to design what we think the ideal reprogramming medicines might look like. We can prompt the system the same way you would another generative type of model. We might ask it to say, what's the ideal group of transcription factors that would take me toward a younger hepatocyte or liver cell? We can then have the system generate a number of hypotheses, test those hypotheses in experiments, and retrain the model in a virtuous cycle. And here's actually what that looks like. This is one of my favorite videos that's ever been generated. That's in the world, by the way, not just like here physically. What we're watching is one of these systems navigating the hypothesis space of all reprogramming combinations. On the left, what you're looking at are the transcription factors the model is choosing to test at each cycle of the experiment in an embedding space. So here, points that are closer together are TF combinations that have more similar biology. And on the right, what you're watching is the performance of the very best hits the model has discovered over each cycle of experimentation. Something quite funny that I noticed when I watched these films is that the model learns how to reprogram cells similar to how humans did, but much faster. Humans first learned how to reprogram cell age and identity together, which is exactly what happens in our model. It can make old cells look young before it figures out how to do that without modifying their age, which takes many more cycles of experimentation. Now, this is just one of those discovery campaigns, and we can run these in serial, so we can ask the question, how much better is a system like this at prioritizing what to do than a human? And so here, I have the unfortunate role of being this blue baseline. This is roughly how many hits you're going to discover if a human tries to guess what experiments should you do next. By contrast, the AI systems our team has built are now far superior to anything at least I would be able to guess. So we've already really achieved superhuman performance for this type of experimental design. Every dollar we invest, we're able to get more discoveries back out. We also have confidence these models will continue to get better over time, even if we were to freeze all of our algorithmic progress today. That's because we observe very common scaling phenomena here, the same way that have existed in natural language and computer vision for many years. As we gather more data, the performance of these models improves. So even if we just keep running the same cycle we've been running historically, we expect we'll end up with better models a year from now than we have today. Perhaps more interestingly, we also see some of these same scaling phenomena in reprogramming biology. As a therapeutics company, what you really care about is not how many potential drugs you have sitting in the freezer. That's interesting, but it's not necessarily going to help you build a great business. What matters is how good the top few drugs are that you take into the clinic. So really the quality of the best things you've found so far matters more than just the raw quantity of interesting phenomena you've observed. And here, what I'm showing you is that we found, as we discover more hits, more things that make an old cell look young, the quality of the very best things we've discovered goes up over time. This is exactly what you'd expect if the quality of some of these potential reprogramming medicines was distributed as a power law, the same way the quality of the best companies might be. Now, one might think that this is just a toy technology or an ancillary bet within the company. And what I'll try and convince you of is that actually these tools have made really meaningful impacts in the business already. We've taken bigger bets on generating data year over year, so we've continued to generate a larger corpus, even though we're already far ahead of where the rest of the world might be. And those bets have continued to pay off. Every year as we gather more data, the performance of these models improves, and the hit rate in our screens goes up. As we test more combinations of transcription factors, the likelihood one of them actually works has been getting better and better over time. And what that means is that the cost of finding a group of transcription factors that makes an old cell not only look younger based on the genes it's using, but act younger based on how it functions has gone down precipitously. And of course, when something gets cheaper, you buy more of it. So we have found far more combinations of these reprogramming factors that are able to restore youthful function than we did in prior years. Now, given how successful this discovery process has been for us, in the past year, we decided it was time to diversify our bets across the unique biologies. We've historically had a couple therapeutic programs, and it felt like it was the right time to bring on a third. This has a couple benefits for us. One is that we're taking more unique biological bets, and another is that in the success case, we have the opportunity to actually treat a larger diversity of the pathologies that arise with age. Today, we have 3 therapeutic programs active. Our first was our program in metabolism, where we're focused on restoring youthful function in hepatocytes, a fancy Greek word that literally means liver cell. I hate how we do this as biologists. We have another program focused on restoring youthful function in T cells to treat things like inflammatory diseases. And in this latest program, we decided to focus on the vascular system that permeates all of our other solid tissues, and in particular, the endothelial cells. These are really the walls of the pipes through which all your blood and signal and circulatory system flows. Now, this might seem like kind of a weird choice. Endothelial cells are not necessarily one of the first things that pops into your head when you consider your own aging biology, at least not the first thing that popped into my head. But I think it's actually a really huge opportunity because these cells are everywhere. They're in every other tissue. And that means that their dysfunction and their age actually has been implicated in a role in the decline of many different tissue systems. Very prominently, there's a clear indication that the aging of your endothelium leads to a loss of function in your kidneys, which has all sorts of negative downstream consequences. But it doesn't stop there. There's also a role in the heart, a role in your liver and your metabolic system, and even some very compelling evidence that breakdown in your endothelium is one of the first stages in cognitive decline in the beginning of neurodegenerative disease. So we're optimistic that if you could restore function, even in just this one cell type, you actually might be able to have really beneficial effects throughout the body. Now, if you want to study aging in a given cell type, the very first thing you have to do is actually get a bunch of cells from young and old people in a given tissue. And it turns out that because not many people study the endothelium, there's not a catalog or like a store you can drive up to and buy these samples. And so in order to even begin this investigation, we had to vertically integrate and begin acquiring organs ourselves. So we've begun to receive research organs that can't be transplanted, unfortunately, for various circumstances. And in order to make use of these generous donations, we can procure them for research purposes. We can then isolate endothelial cells from human kidneys, and we've built out a biobank where we have cells from dozens of young and old human donors. To our knowledge, this is the largest biobank of its kind, and so we've had the opportunity to ask, for the first time, really even some pretty simple questions. Are endothelial cells in old kidneys worse than young ones? Until you have this material, you can't even really begin to run that experiment. So now that we have all those cells, and I'm alighting over a lot of late night phone calls about kidneys being shipped, a lot of Saturdays picking things up off the loading dock, you now need to actually start testing transcription factor sets and asking, do any of these make an old endothelial cell look younger? When we first started the company, this was a crazy, painful process for every cell type. When we first built out the tools, it took more than a year just to get a single screen to work in the first cell type we tried. We got better at it over time, but this was really the first time in the company's history where we were able to bring on a new cell type and open up the aperture of opportunity without having to build anything new. Everything just kind of worked when we ported it over. And that tells us we've reached a state of maturity where the technology is pretty general. We're not over-indexing on cell type X or Y, but rather have the tools we need to get to a broad diversity. Now, the experiments we run are actually pretty simple in concept. Once we have a bunch of those old endothelial cells in a dish, we can introduce many groups of transcription factors into them, all at the same time. Thanks to the stochastic nature of life, every cell gets a different combination, and you can test all the combinations within that pool together. We then turn those genes on and off the same way we would when we deliver a drug, and we measure what happens to the genes the cells are using and how regenerative those cells might be. Because this worked pretty well, to our knowledge, we've now generated the largest data corpus of reprogramming in endothelial cells. And from that data corpus, we've already began to make early discoveries of payloads that are able to make an old endothelial cell look like a younger one based on the genes it's using, and potentially more importantly, act like a younger one based on some of the functions we can measure as well. Now that we actually have some hits we're excited about, you need some way to test. Can I make an old cell act like a young one in addition to just look like a young one? If you don't have a tool to measure the function that changes with age, you're certainly not going to be able to fix it. And so we've built out a series of these functional assays. Here I'm showing you just one. We found that very prominently, old endothelial cells from human kidneys lose the ability to regenerate compared to their younger counterparts. This might sound obvious to those of you who have, experienced aging in your own lives, but surprisingly, this has not actually been measured before, likely because no one's ever had this many cells in the same place. So this gives us the tool we need to be able to ask the question, can I make this old cell act like a younger one in the first place? We've also developed systems that allow us to do this in vivo in animals in the context of all the complex interconnectedness of the body. So one way we can measure how these endothelial cells are functioning is by measuring the function of the kidneys themselves. You can do this in a pretty simple way. You can deliver a fluorescent dye to an animal and then use a really special little camera and measure how well that fluorescent dye is filtered out of that animal's bloodstream. If it's filtered out quickly, like it is in the young animals, you've got good working kidneys. Unfortunately for these old mice, their kidneys don't filter nearly as quickly, and this mirrors exactly what's happening in humans. So now we have yet another system where we can deliver these payloads and ask, is this working? Are we actually making the old cells function like young ones do? We can take that even a step further and demonstrate that some of that dysfunction is very specific to the endothelial cells. We can challenge these kidneys in a way that we know harms really only the endothelium. And what you find is that the young animals are perfectly resilient, whereas the old animals are far more susceptible, suggesting those endothelial cells can't really become as resilient to damage as their younger counterparts are. Now, we've got two ingredients in our recipe for finding reprogramming medicines in endothelium. We've got a system we can use to discover the payloads. We've got some assays we can use to figure out if they do what we want them to do functionally. But there's still a missing ingredient on the table, which is in order to actually build these medicines or even test if they work, we need some way of getting these reprogramming factors into the cells we care about in vivo. So we need some what we call delivery chemistry, some way of transporting RNAs that encode RTFs to the right cell type at the right time. This didn't exist off the shelf when we started the program. We just kind of made a bet that we'd be able to get to a solution. And thankfully, our team delivered. We tested a whole bunch of different approaches, and we were able to find a chemistry that delivers these reprogramming factors at remarkably high efficiency. To our knowledge, this is the most efficient delivery of these cells anywhere in the world right now. And we think that this now gives us all the components we need to actually run that recipe a few times. And hopefully a year from now, we'll be able to report on the results. We'll be able to show you that we can rescue aged function in a tissue. Now, once you've run a program like this for a while, the ideal result is that you actually have a medicine you can hold in your hand and eventually carry forward to human clinical trials. And we've talked a lot about what these medicines are, and you might be wondering, like, what does that actually physically look like? Like, what does this composition actually do? And in other companies, this might be the time where I pull back this curtain and show you a cool robot, maybe like a roadster spinning on a screen. I might land a rocket outside. In molecular biology, after you burn millions of dollars, what you get is a tube that looks like this, filled with our preclinical candidate, and it's mostly a clear liquid. So unfortunately, I don't have such an impressive demo, but I appreciate you guys letting me get away with a gimmick. It's a very, very valuable tube. And what's more important than how cool it looks, at least is what I tell myself at night, is what's inside. And what's inside are special lipid nanoparticles. These are really a bubble of fat that carries whatever is inside to cells that take up that fat like a Trojan horse because they think it's a nutrient. And inside are RNAs that encode the transcription factors that make an old cell act like a young one. So it may not look like much, but the technology is far more powerful. Now, how do we actually make this bold claim that we can make an old cell act like a young one? What is the data that gives us conviction this is true? For us to claim this, we believe that not only do we need to make an old cell better at just one thing, we need to make it better at everything that is impaired with age. And so we call this pleiotropy, another fancy Greek word which literally means do a lot of stuff. And so what I'm going to walk you through are a few pieces of the evidence that this payload that's in my hand does a lot of stuff. The first piece of this evidence is we can actually demonstrate that we can make old cells regenerate like young ones do. If you cut an old liver in half, it does not regenerate very well. But young ones, miraculously, like Prometheus on the rock, heal perfectly in about a day. We can close that gap by about half with just one dose of this reprogramming medicine that I'm holding. Now, of course, regenerating after being cut in half is not the only thing that your liver does, at least if you're like me. Tell me about your lifestyle later if that's not true. But many of us can probably relate to this second function of the liver. Aged cells actually suffer damage when you eat high fat or alcohol. And young cells, it turns out, are incredibly resilient to this. And so we've also found that when we reprogram these aged liver cells, they become resilient the same way that young liver cells are. So this demonstrates to us we're not just rescuing one function that is impaired with age, but multiple, in the same way that making an old cell young would be expected to achieve. Now, that demonstration I just showed you is in a really acute setting. Think about that as a model of going out for a night. What really matters when we think about trying to treat patients is modeling more severe disease that develops over a longer period of time. And here, we can continue to provide these animals with a damaging diet, and unfortunately, the aged animals would naturally succumb to mortality. And again, just an individual dose of this reprogramming combination at the beginning of a multi-month period can help them survive better than they would otherwise, suggesting that this is a real profound benefit that you can measure even without sophisticated tools. Now, none of that efficacy would be meaningful if the drug wasn't safe. Through the Hippocratic Oath, the first thing you need to worry about is doing no harm. So how do we demonstrate safety? Turns out it's actually about as simple as you'd think. You give an animal this drug over and over again, way more frequently than you would to any human. What we found when we've run these experiments is that, if anything, we make their livers healthier rather than sicker, which is the first thing you always check for. And perhaps more importantly, we don't see any evidence of pathology or tumors forming in the tissue. This is what you'd worry about if you were changing cell type. And so the fact that we're getting a clean profile here suggests we achieved the goal that Brian laid out. We successfully decoupled reprogramming cell age, getting these benefits, without actually having meaningful impacts on the cell's identity, which would cause tumors. Now, there's one more phenotype that I hid from you for a moment, and I saved it for last, not because it's most important, but because I think it's actually the most visceral. And I think it gives you a good intuitive sense for how potent these medicines can be, even if it's not the thing you would measure as your endpoint. So what I'm showing you are some animals that have all just had a drink of alcohol. And the only difference between them is on the left, these animals are young, and they are out looking for their next adventure, wondering what's up next, where they're going after this. The mice in the middle are old, and when they have a drink of alcohol, they pass out for 8 to 12 hours, and they're severely sedated. Some of you may have experience with similar phenomena. Now, the mice on the right received a dose of our reprogramming drug well before they ever received this alcohol. And I'll let you judge for yourselves which animals they resemble. I, for one, would much rather be in that far right-hand cage. So again, this isn't the most critical phenotype. It's not the thing you're going to put as the endpoint in your phase 3. But I do think it gives you a really clean demonstration of just how powerful the medicines can be, and a holistic representation where you can see this with your eyes and you don't need any statistics. Now, you might be wondering as well, do we just have one medicine? Like, have we been working all this time and that's all we got out of it? And in fact, we've actually been able to reproducibly discover combinations that have these effects. Here I'm showing you data from just one other combination that we found that very similarly can rescue multiple youthful functions, can make old cells regenerate better, more resilient to alcohol, and even clear fat that accumulates in the liver. This final figure on the right with all those red dots I love because, again, you don't need any statistics to analyze it. The fat dyes red. When you give the animals the drug, which are the photos on the bottom, the fat goes away. I don't probably have to convince you of that too much. Now, just like the payload before it, this payload is also safe when you repeatedly dose it. So it suggests that we're able to repeat this success where we're able to decouple reprogramming cell age and cell type yet again. And in case you're wondering, yes, it also cures mouse hangovers. So with that, I want to turn this over to my co-founder, Blake, who will tell you about how we take these early research results and actually turn them into medicines for human beings.
All right, so we got to move that vial and the LMPs with mRNA in them from Jacob's hand in the lab into the clinic and then eventually into any patients that need them around the world. And one might naively think, well, the good strategy for doing this is you just give it to a bunch of people and see how they age. The problem with that strategy is it requires a lot of people, a lot of time, and a lot of money. And so a more efficient way to show that your drug works is really thinking about the point in the presentation earlier that if you can successfully reprogram the age of a cell, it confers a lot of capabilities on that cell, a lot of youthful functions that it had lost, and you can treat lots of diseases with it. So we can copy the playbook that Eli Lilly and Novo took with GLP-1s by starting in narrower patient populations that are higher burdens of disease, where you can show clinical benefit in a much shorter period of time with a smaller group of people. And so that's what we're going to do. With GLP-1s, they started in a narrower group of diabetic patients, and then they eventually expanded to obesity. And now it's really a drug being taken in a preventative way by a much broader group of people who aren't classically diagnosed with any disease. For NewLimit, we're going to start in narrower indications of aging-related diseases. I'll go into what those are, and then eventually expand to much broader patient populations. So we go through our 3 different franchises that we're working on. One is immunology. Our sentinel or initial indication there is going to be arthritis. And then from there, we could eventually expand to other forms of infectious disease prevention. Our liver program or metabolism program will start in narrow indications of liver disease. I'll go into this a little bit later, but one of them is alcoholic-related liver disease, and then we could expand to broader patient populations like metabolic syndrome. The vascular program is going to start in a condition called chronic kidney disease, and then could expand to cardiovascular disease or even cognitive decline, as Jacob mentioned earlier. And just to remind you all, the point of all this is not to, maybe we'll have one drug that works. The point is that taking all of these together one day, if we're so lucky, will allow everyone to live healthier lives for longer. Okay, I'm going to drill in a little bit on just technically how we would approach this for our liver program. So we'll start in alcoholic-related liver disease. This is unfortunately a really challenging disease for patients to get. It's less than 50% of patients diagnosed will make it past 5 years. And there's effectively no useful treatments on the market for these patients. It's reasonably prevalent, with 2% of Americans suffering from ALD and 30,000 deaths a year. To put it in perspective, if all we did was develop a drug for ALD, it could be worth $20 billion on its own. But we believe in the ability for these to expand to much broader patient populations. So an example of that on the liver franchise would be metabolic syndrome. Metabolic syndrome is experienced by over 50% of the people over 65 years old around the world. On that expansion alone, it's worth many multiples of the $20 billion ALD franchise. How do we develop that? Well, you've heard a little bit about it today. We've been busy upstairs and in the bottom floor here, both training the AI models, doing research and development, and now producing the actual drug product that's going to go into these clinical studies. And I'm excited to announce, we had a tease earlier, that we'll be putting the first age reprogramming medicine into human patients next year. All right, now we're going to drill in on the vascular program. Kidney disease is basically irreversible, just like ALD. There's no effective therapies available for patients. It actually has a really high burden of disease. 14% of people that are older have CKD. Over 95% of people over 65 have some kind of kidney impairment. As a sense of scale, with dialysis and the amount of burden on the healthcare system for people, for those taking care of people with chronic kidney disease or other forms of kidney disease, it adds up to 0.5% of US GDP. The value of just developing a CKD drug is something like $60 billion. And just like we talked about with the liver program and our other programs, we believe there are clear expansion opportunities going from just CKD to then eventually broader kidney dysfunction in the elderly to potentially even cognitive decline as you maintain the blood-brain barrier as people age. What is the value of being able to prevent cognitive decline? Some crazy multiple above just the CKD program alone. And that's what we're going to be going after. How do we do this? So the vascular program, we actually just launched last year, basically less than a year ago. Why do the timelines look a lot faster? The underlying platform that we built, that Jacob and Brian talked about, from everything from our Write program, where we insert these genetic perturbations, these combinations of transcription factors, to our Read program, where we read out the profile of these cells in single cell, to our Predict team, which is the bioinformatics and machine learning to suggest what experiments to do next and select what to move forward. All of that keeps compounding, so we can add a new cell type and achieve results much faster than any of our previous programs. And so we're compounding on that, and we're predicting that within just a few years, we're going to be able to get this medicine into patients and be able to help them from just a cold start a year ago. All right, so building this company, building any company, it's a stepwise approach. What is NewLimit going to look like next year with these plans in place? We'll have our first age reprogramming medicine in human clinical studies. We're also going to launch another cell type program. So we'll have 4 franchises underway after immune, vasculature, and metabolism. And if we look out just a few years, we plan to have 3 drugs in human clinical studies. We'll be working on 5 or more tissue cell franchises alongside our 3 today. And that will 3x our discovery rate efficiency. And what do we mean by that is we not only want to decrease the cost per discovery, which we are maniacal about doing and tracking, but also increase the quality of every drug we move into the clinic. And we think we can increase quality by 50% while having the cost of developing such a program. Next, I want to thank our sponsors. It's like easy to take bets on companies where you think, like, hey, revenue next year is going to be higher than this year. And it takes more conviction to bet on companies with longer term legible readouts. But I don't think they're that crazy, because really companies like NewLimit are the ones that can potentially bend the arc of humanity in a multi-generational sense, and not only just improve the species, but improve all of our lives in a really substantial way. And so I want to thank all the investors on this slide for participating in it. But now the most important slide is the team. We're up here presenting all the work that's really an output of all the people here on the slide, some of which are in the room right now. And I wanted to double down on emphasizing this point. It all sounds very, oh, you're doing aging research, but it's all done by the people on the slide coming to this building every day. We basically do everything in-house. So kind of the frontier of humanity's progress on aging medicines is on this slide and in the room with you. And what I want to encourage you to do is come join us on working on it. As crazy as aging research is, we are actually most bottlenecked by people, by talent joining the team. And so if you can't come for work at NewLimit for some reason, which I still challenge you on that, I want you to send us the name of somebody that you think would be really good to work at NewLimit. And so you have a careers thing there, or you just come and find anyone with a NewLimit shirt on, or Brian, Jacob, and I afterwards, and tell us their name, and we'll go hunt them down. Okay, so with that, I want to invite some of my colleagues on the stage and my co-founders back up, and we're going to do some Q&A.
So the unique thing about the Yamanaka factors, right, is that they work generically in any cell type. Now that you have these 3 campaigns where you've discovered things that are effective in 3 different cell types, you start to see any points of convergence where you are finding the same factors recurring, or that would suggest something that's kind of more generalizable, or does it end up being very context specific?
As of today, we think it's going to end up being very context specific. But that said, we really looked at an N of 3 cell types, all of which are fairly distinct from one another in terms of their chromatin state. They develop in very different ways. My suspicion would be that if we start to look at cell types that are a bit more related, say T cells and B cells, which share a lot of their developmental lineage, then you'll start to see that there can be shared programs, and that discovering a payload in one might get you part of the way to the answer in another. But as of today, it's ended up actually fairly distinct. That might sound initially kind of disappointing, oh gosh, you got to crack all these passwords. There's not just going to be one. But I think there's actually a hidden benefit to it as well, which is that when you deliver these therapies as actual therapeutics, you can localize them to your desired cell type of interest, but some of it goes other places. You never have perfect delivery specificity. And the fact that these payloads largely have an effect in one cell type and not others gives you an additional layer of safety. If a payload intended for the liver doesn't do much when it goes into a blood cell, that's actually a good thing for you in that circumstance.
I won't ask you a question about the meaning of youth. So maybe a slightly more kind of technical one about delivery. If you want to go after all endothelium, right, which the idea is, okay, these cell types bathe every organ in the body and we can have systemic effect, but you're using LNPs, how are you going to get them to every tissue in the body?
It's an astute question, and one that does not involve epistemology. So many of these different endothelial beds, as they're called, have slightly different physical properties, and they also do express some different markers on the surface of the endothelium. And so for our initial products, we actually plan to target them to just one of these beds at a time, as opposed to trying to go everywhere. That potentially gives you the ability to have really dramatic efficacy in that tissue without having to worry quite as much about the broad safety risks. And you can achieve that specificity both by tuning the specific parameters of the particle, even stuff like how big it is determines where it goes, and then also having it bind some of these moieties that are on the surface of some endothelial beds but not others. So that's how we can get localization to, say, the kidney but not the heart. The other opportunity you have for making it specific is you can even do local administration in some circumstances. You can deliver, for instance, for the lung through nebulization in a way that isn't exposing it to the rest of the body. The hope over time would be that if you're able to demonstrate safety and efficacy in some of these tissues separately, that then you get to a place where there's just one formulation that maybe has pan targeting, and you get to a place where that one formulation addresses many of these pathologies at once.
When can I try the medicine?
I got one in my pocket for you. Upstairs. Don't tell anybody.
I wanted to ask about cracking. How many passwords have you cracked? So I think on one of your early slides, Jacob, you mentioned that you had something like 30 or more like prototype medicines. And then you talked about 3 therapeutic areas. I'm curious how much overlap there is or whether that's going to, unlock new. Are those passwords for new diseases or a deeper pipeline for those 3?
Can I nominate you?
Sure, I guess so. I do think those 30 at least, they tend to be for a wide variety of diseases. But we do see that what does end up happening for particular advantageous or rejuvenative therapies in one cell line tends to be unique there. But what's nice is we're trying to extrapolate how a perturbation that affects a certain gene expression phenotype translates into each unique mechanism. So by looking at those different mechanisms, we can take each of those 30 and assign it to the wide variety of things that we think it may do, and then get the relevant sort of functional assays that can test it in that domain. So it's both, yes, it will hit a large number of things, but some of them will actually be quite specific for certain indications. So it's fun digging into that.
Let's say everything goes right, like really right, over the next 20 years. What does the world look like?
Well, I think the societal implications are slower than people expect. Because it's just like you slow down the rate at which people develop disease, you slow down the rate at which people age. And so it takes a long time for that to play through on a societal basis. So even like in 20 years, if the drugs are out, it's not an immediate change. Like people feel younger, they feel healthier. But then as you keep running the clock forward, all of a sudden you have lower cost of care for people. People can live longer lives. That probably starts to change things more dramatically, but it's a slow compounding effect. It's not like some immediate switch and all of a sudden there's 2 times more people living in the world.
Yeah, like just to build on that, like a wild success case would be we treated 1% of humans on the planet in 20 years or something, and that even that alone wouldn't probably have dramatic effects across society. But sometimes people are worried about like overpopulation or something. Well, we're seeing actually birth rates decline in most developed nations, so I don't think that's as big of an issue. The other thing people bring up sometimes is that, there's kind of this saying like science advances one funeral at a time. And, sometimes as people get older, they're, in our government or whatever, like they become, wedded to one particular way of thinking and their brains are not as like open to new ideas. And so sometimes people think of this as, well, like, there's not going to be any turnover of new ideas in society. I think that could be true if you believe that these kinds of drugs won't apply to the brain. But if you had the mental acuity or plasticity of a 25-year-old again, maybe older people would become open to new ideas. Like it could be a very different world. So
that's the part I want to hear about. I'm not asking you to go through like some moral struggle session or something like that. I want to know like, Fred is not 38 anymore. He's 58. And like, how do I feel if I'm like on the full suite of NewLimit over here?
Well, the intellectual honest answer is we don't know exactly, but the potential is that, I mean, how did you feel when you were 25, right? You probably went to the gym. You didn't feel as tired the next day.
Unfortunately, I didn't because I was working on Coinbase. But
I think, like,
you could be back working at Coinbase at 50.
Exactly.
Yeah. Energy, mental acuity, metabolism. I mean, just generally like aches and pains as we get older. I mean, all kinds of things could be much, much better.
I think also it's going to be a jagged success. So like even in 20 years, if everything goes right, we're not going to solve every tissue, every cell type possible within 20 years. If everything goes right, we've made a big dent. And hopefully we've picked correctly where even just some of these, like even if we just get metabolism right, you probably like can feel the benefit of just that. And if we can go through a few of these, I think you can like genuinely feel much younger than you otherwise would have been at that age, and in a very material way. I mean, some cell types that are on our list that we're not going to talk about now, like it would improve mobility and quality of life in ways that are outside of the ones on this list. And I think this is like a very reasonable, like possible future. It's not just like adding 1 year of life at the end of your life. I think everything that we've found so far at NewLimit suggests that aging is much more plastic than any of us realized going in. And that even if we're not the ones to achieve it, like someone's going to, and aging is much more mutable state. And like aging drug, like the TLDR in NewLimit is like aging drugs are coming.
If I can make one more addition to that, I think the implications are interesting, but it takes 20 years to get a human being kind of trained up. Like you're sort of taking from the world until you're like a certain 20 years in or so, and then you can start to contribute in your first job or something like that. Maybe if your PhD program goes later, but and then you have
taking arrows over here, man.
That wasn't supposed to be a joke. Sorry, but then you kind of have like. Most people have the power zone of their career for the next 40, 50 years, if you're lucky, right? And then, we start to slow down a little bit and move into retirement. And so it's kind of like there's only maybe 40, 50 years that in a good scenario, we have people, driving civilizational progress, right? Other times our bodies give out on us, our minds give out on us. We find other priorities. And so, if you imagine somebody someday could be 100 years in the power zone instead of 40, we'd have just people with much more wisdom, experience. The implications for GDP, like all kinds of things, are pretty profound if you think about that.
Instead of retiring, you too can go back to Coinbase. Okay, so next question.
Amazing talk, super exciting. I'm curious about the durability. So like how long before these mice go on a bender can you administer a drug and it will still work?
So the longest term data we have today are some of those survival data we showed you where there's benefit even months after an initial dose. We imagine that the medicines, when we first go into the clinic, will be dosed something like monthly. We don't know the exact number yet, but that's the right order of magnitude. Over time, there's lots of reasons to believe that these medicines could be much more durable and several different paths to get there. To give you just the short version of that, the sort of ultimate durability existence proof is that epigenetic reprogramming is effectively how all your cell types remain different cell types. That's why your eyeball, at least my eyeballs, don't spontaneously become pancreases. So that tells you that these marks can last a whole lifetime. And then others have demonstrated that you can reprogram these marks and actually make them last years in primates. Now, in order to actually make these medicines more durable and get toward that upper bound, you can either find payloads which are inherently more durable, you can use pharmacokinetics like engineering the mRNA molecules to make them last longer, or you could get to a place where maybe they're not more durable in principle, but the dosing format is very convenient. If you could get to a place where you administer a reprogramming medicine the same way you administer Ozempic, maybe it doesn't actually matter if it lasts multiple months because it's just pretty easy to redose. And so we think there's several orthogonal ways to solve the problem, and we're pretty optimistic we'll get there over time.
How do you guys see NewLimit fitting into a future where perhaps, the regulatory constraints around having to get approved for a particular indication are no longer there? Do you guys transition to, just targeting aging directly or however that's defined? And how do you guys, think your drugs would fare in a world where people are developing drug against that central aging indication?
I think there's a few different tacks here. One is the outline that we had in our slides where we start in sentinel indications and label expand up. I think there's other ways that we could foresee either the US or other geographies having a different posture towards more preventative medicine kind of drugs, which right now are, really long trials are almost prohibitively expensive to run. And I'm actually pretty optimistic given the work that we're doing, the work that others are doing, that there's more and more of a press on that. To give you a sense, today, if you're running a clinical study, you really take on the cost of care for the patients in the clinical study. You can imagine a world where it's if you're to really make preventative medicine happening, if insurers just covered the normal cost of care for those patients, you cover severe adverse events. Now you dose them. You still have to follow them for a long time. There's no way to get around the human biology of it. You just drop the cost of that trial by maybe 90, 95, 99%. And so that's an example. And I think there's many others. And both us and others are going to participate in a way that drives to measurable outcomes that we think like demonstrate that it's really beneficial to patients, but also unlock this and enable it and enable access for everybody.
So since all these diseases are happening at the same time, how much does health span actually improve if you cure aging in one organ?
I'll take this because I'm not seeing any eager takers because it's a hard question to answer. The short answer is we don't know, but we can extrapolate from a few different pieces of data. I think directionally it's clear that rescuing even a single organ can add multiple healthy years to your life. One of the most extreme pieces of data that one can use to answer a question like this is actually looking at patients who get transplants for different organs. And so if you look at patients who, for instance, get a transplant for their liver, patients who receive a young liver rather than an old liver are way better off. They live longer, they have lower rates of diabetes, and many other positive sequelae. So it suggests that even if the rest of the body is old, having a really healthy, youthful tissue, even just one tissue, can provide some pretty meaningful benefit. And then I think you start to get some nonlinear interactions as well. If your liver was healthier, your kidneys were healthier, your heart was healthier, potentially that adds up to more than the sum of its parts. There's no clean experiment that can give you that answer today, but I think the data are directionally positive. Okay, one final question from Will. I didn't tell him to do the last question, and then we're gonna head upstairs.
I mean, it's related to what you just said. I mean, you're obviously very focused on reversing aging phenotypes, but presumably also these TFs are just modulating physiology broadly, and you could find things that like improve young physiology to, superhuman levels. Have you looked into any of these things or thought about it at all?
We have yet to make the superhuman mouse. We have made all sorts of weird stuff, though. When you screen lots of combos of TFs and you start looking at the cell types that emerge, the genome supports a much broader set of stable states than you think it does. We don't spend a lot of time on those things, but if you're like a weird cell connoisseur, we should chat afterwards because we've probably made something that'll suit your fancy. Awesome. Thank you all again so much for coming. This has been really wonderful to spend this time with you all. Let's go head upstairs. There's a bar. If you look hard, there's NewLimit 1001.